We are all familiar with enzymes and their importance within the body, but did you know that only slight changes to the enzyme's environment can actually cause it to stop working? My name is Alyssa, and in this video we will investigate those different environmental changes and see how they affect the activity of the enzyme. We'll start off by defining enzyme activity, and then look at how temperature, pH, and salinity can each affect this enzyme activity. After watching this video, you will be able to identify what factors affect enzyme activity, and you will be able to answer questions that ask you specifically what conditions are optimal for certain processes. The terms enzyme activity, enzyme velocity, and enzyme rate are all used interchangeably on the MCAT, so make sure that you can recognize each of these terms. Enzyme activity measures how much enzyme is present in the active form, meaning how much enzyme is present that can transform substrate into product per minute. Here we have two substrates in the active site of an enzyme. This enzyme is active because it is able to convert these two substrates into two different products. There are a few different factors that we must consider in order to determine if an enzyme will be active under different environmental conditions. Let's take a look at a few examples of these. Temperature has both a positive and a negative effect on enzyme activity. A positive effect is that the rate of reaction doubles for every 10-degree increase up until a certain point. Within the body, this point is about 37 degrees Celsius. However, the negative effect is that once enzymes reach a certain temperature, the activity will begin to decrease as temperature is increased. This is because at high temperatures, enzymes can denature due to the increased molecular motion caused by adding energy to the enzyme in the form of heat. In some cases, decreasing the temperature and therefore the molecular motion will allow the enzyme to refold and regain function once again. Another factor to consider when thinking about enzyme activity is the pH. Enzymes are made to function best in specific pH ranges. For example, the pH of blood is 7.4. Therefore, enzymes meant to function within the blood must have their highest rate around this pH value. Altering the pH to values higher or lower than 7.4 will greatly affect the rate of reaction. This is because changing the pH can protonate or deprotonate the enzyme, affecting hydrogen bonding between amino acids within the enzyme. This alters not only the active site, but the entire enzyme structure as well. Let's look at a few physiological examples of enzymes at their optimal pH. As mentioned before, blood has a pH of 7.4, and so enzymes meant to function within the blood should have a maximum rate around this value. In the stomach, enzymes such as pepsin must be able to function in the high acidity of the stomach, and so these enzymes have optimal pH values around 2, just like the pH of the stomach. Finally, in the small intestine, pancreatic enzymes must be able to function in the alkaline environment, and so their optimal pH value is around 8.5. The last factor to consider is salinity. However, it is important to note that these effects are only visualized in vitro, or in tests within the lab, and are not of physiological significance. When the salinity is altered, it introduces ions to the solution, which in turn can partially affect the hydrogen and ionic bonds that hold the enzyme in the proper conformation for activity. Let's say, for example, we have this enzyme that takes two substrates that react in the active site. If we increase the salt concentration, this can alter the conformation of the enzyme, and the blue substrate can no longer enter the active site and react. Now that we have seen different ways to alter enzyme activity, let's try out this practice question that asks about the optimal pH for an enzyme involved in digestion. The answer is A. Chemical protein digestion begins in the stomach, which has a pH of 2. Therefore, enzymes required for this process must function optimally at this pH. Good job if you got that question correct. In this video, we examined 3 different ways in which enzyme activity is affected within the body. By increasing the temperature only 10 degrees, we can double the rate of reaction. However, within the body, after reaching the optimal temperature of 37 degrees Celsius, activity decreases as temperature increases. We also examined the optimal pH for certain enzymes to function. 7.4 for enzymes in the blood, 2 for enzymes in the stomach, and 8.5 for enzymes in the small intestine. Remember, if the pH strays from the optimal value, the enzyme activity will decrease. Finally, we looked briefly at salinity and saw that this only affects enzyme activity in vitro studies. Thank you for watching this video, and good luck studying for your MCAT.